Cloud. Hello, everybody. Okay, chapter 24. Okay. Okay. So overview. So let's go through the immune response. Okay. So we'll focus on innate versus actually, I'm gonna focus a little bit on communication and then innate immunity and then adaptive immunity. Okay. All right. So the immune system has three major functions. Yeah, hold on. Actually, probably sharing the wrong screen. Yeah. Okay. So one. Okay. Um, okay. So immune system tries to do three big things. One, it tries to recognize and remove abnormal cells. So like cancer cells. Okay. It removes dead or damaged cells because dead, well, not dead, but damaged cells can eventually lead to cancer cells and not removed. Dead cells become a problem because normal cells can't grow in the same location and they can potentially harbor disease-causing pathogens. Number three, so remove disease-causing pathogens from the body. Okay. Okay. So, okay, there are barriers that must be penetrated first. Okay, and then if the barriers actually penetrate, so the physical, mechanical, chemical barriers, you should know what they are. Okay, we went through that anatomy, and then innate immune system, and then adaptive immune system, and then so okay. So if you've taken physio before, this chapter is not physio. This is physio. If you've taken microbiology before, this chapter should be easy peasy because we actually go into more depth about the immune system for physio than or in micro than physio. Okay. Okay. So you should know the difference between primary lymphoid tissue and secondary lymphoid tissue. So primary lymphoid tissue is going to be where the T cells and B cells develop. Secondary lymphoid tissues are where they work, okay, where they function. Okay. Okay. So white blood cells are also called leukocytes. So you should know what basophils and mast cells and um, neutrophils are. Okay. 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 These guys are these three basophils, eosinophils, and neutrophils are granulocytes, which means they have granules in them, and that means they can release things from those granules. Okay, so basophils and mast cells, they cause inflammation and they act in something called an innate immune response. They cause inflammation. Okay, they're technically the same type of cells. Basophils circulate, and mast cells are the fixed in tissues. Basophils are by far the smallest number in the actual blood of all the immune cells because they function mostly as mast cells. Okay. Okay. Eosinophils are going to be involved in parasites and allergic reactions. They actually actively destroy parasites like little worms and stuff that get into your blood. Okay. And then neutrophils are the most common, and they're going to release cytokines and inflammatory mediators along with being phagocytic, which means they'll eat the bacteria. So neutrophils are actually going to be the first thing that arrives at the site of infection, and they're going to eat up all the or they'll try to eat up all the bacteria. Sometimes they succeed, sometimes they don't. Okay, but they'll try to eat up everything, and they're going to release cytokines to trigger the rest of you, bring the rest of the immune system in. Okay. Okay, monocytes. Okay, are turned into macrophages. So monocytes float through your bloodstream, and when they get to the tissue, they become called macrophages. Okay. Okay. Monocytes have different names in different places. Kupffer cells in the liver, microglia in the brain, osteoclasts in the bone, dust cells in the liver, and lungs, etc. Okay. Dendritic cells are the antigen presenting cells in the skin and various organs. And last but not least, lymphocytes. They're derivative of they're derived cells, and we'll talk much more about those in a little bit. Okay. So the development of immune system cells. Okay. They require cytokines. Okay. Called interleukins. Interleukins in between white blood cells. Okay. All right. T cells and B cells. Okay. T cells, they are made in, so all immune cells are made in the bone marrow. T cells actually migrate from the bone marrow to the thymus to mature, and then B lymphocytes mature in the bone marrow themselves. Okay. Natural killer cells also develop in the bone marrow for the most part. Okay. So lymphocytes mediate the adaptive immune response. So B cells and T cells are not specific, or they're very specific, and they only bind to one particular antigen. So they're not generalists. Okay, is what I was trying to say. All the rest of them are pretty much generalists. Okay. So all lymphocytes that bind to one particular antigen and are clones of each other. So I like to define it backwards. Okay. So when lymphocytes grow, when they actually encounter an antigen, what they're going to do is make many copies of itself to fight off that antigen. And those many copies of itself are clones of each other. And that's what a clone is. Okay. Okay, so you can see they all come from one big pluripotent hematopoietic cell themselves. Okay, everything in your blood comes from there. Okay, so thymus gland, that's going to be for development of T cells. Okay. Okay. So your thymus, okay, what happens in the thymus, the cells that are self-reactive are eliminated. That way you don't have an autoimmune response, or else if there was no thymus, everyone would have autoimmune response. Okay, the thymus does a training to go, okay, look, you're reacting to something that's in the thymus, well, you're dead. So it dies. Okay, the ones that don't react survive. Okay. Okay. Distinguishing self from non-self is something called self-tolerance. Okay. So we went over this in micro. So if you recognize self, then you're destroyed. Okay. Okay. So this is negative selection. Okay. Early pathogen exposures strengthens immunity. This is why like kids should play in dirt and should be dirty and stuff like that, because the good hygiene hypothesis is that people who have good, really good hygiene when they're children end up with children with uh, allergens. They're allergic to stuff. Okay. Sorry, I'm losing my ability to really speak today. All right. Development tolerance. All right. So, okay. So there are some definitions I really kind of want you to know. So the ones I check off, you should definitely know. Okay. Chemotaxins, you should know. Oxidants, pyrogens. Okay. You don't need acute phase proteins, including bradykinin. Okay. Complement, grandsons. Okay. Heparin, you probably already know. Okay. Make sure the ones I check off, you know. Okay. Okay. Page 2 on this list. Oops. Let me change color, make sure you understand. Histamine, okay, interferons, interleukins, okay, lysozyme, back, warfarin, okay, and TNF, okay. All right, make sure you know all those. Where am I? Okay. Okay. So this is a membrane attack complex. Okay. Complement proteins. Okay. They usually float around in the bloodstream, and in the bloodstream, they're usually too low levels to do anything, but they're attracted to antibodies. When there's enough antibodies binding to something, the complement will bind to the antibody and they'll start interacting with each other. And eventually they'll have so many that they actually form a membrane attack complex. So the membrane attack complex will puncture little holes into the cell wall, and that cell wall will no longer function, and that will cause the cell to slice. Okay. Okay. So the major histocompatibility complexes, or MHC, there's two classes. There is MHC class 1 and MHC class 2. Okay. So the difference in MHC class 1 and MHC class 2 is actually their function. Okay, MHC class 1 is expressed by all nucleated cells. So everything except red blood cells, really. Okay, so MHC class 1 molecules tells you about the cell itself. Okay, so if I'm a regular cell, I have MHC class 1. It tells me everything that's inside me, all the normal stuff that's inside me. And if I have viruses and bacteria that get inside my cell, that gets also expressed on MHC1 as well. Okay, MHC class 2 are only on something called antigen presenting cells, or APCs. Okay, they signal not what's inside you, but what to kill. If you present it on a MHC class 2, it's a target of what to kill. Okay. Okay. So these antigen recognition molecules or antibodies and T cell receptors. Okay. So T cell receptors are going to act on MHC class 1, and they are going to kind of check every cell, because every cell has MHC class 1. The T cell receptors are going to bind to MHC class 1 and see if there's anything it needs to kill. Okay. Okay, so MHC class, blah, blah. Okay, so let's talk about antibodies further. Okay. So antibodies. So these are antibodies. There are 5 classes of antibodies, so you should definitely know what these 5 classes are. So, by the way, the mnemonic is MDs. Type it out. MDs give everybody aspirin. Okay, so M D G E A. Okay, you should know what each of them are actually for. Okay. Okay. So the way antibodies work, antibodies have a Y shape. Okay. This end is the antigen binding site. Okay. This end is the FC region. This end, the antigen binding sites, will bind to antigens. Okay. Bacteria, viruses, pollen, whatever it binds to. This end will attract immune responses from the body. Okay, complement protein bind here, macrophages bind here, oxidants bind there. So like anything that attracts the immune system. Okay, so if you have something that collects a lot of antibodies, then the immune system's going to want to pay attention. So they'll be attracted to the FC region. Okay. All right. So pathogens in the body, we'll basically skip. Okay. You should know the differences between bacteria and viruses. Okay. Um, these like these 3 lines of defense you should these things you should know already. Okay. Okay. This one, this slide you should know is innate immunity responses. Okay. Chemotaxin. So when there's cell damage, they release chemotaxins. Chemotaxins, like nitric oxide, attract white blood cells, and they get white blood cells like neutrophils to leave the blood vessel for the tissue, and that's extravasation. Okay. Okay. So you should you can see like this is what happens. The pathogen can bind directly to pattern recognition receptors, or they can have the other thing they can have. They can have antibodies on them. The antibodies on the surface will be attracted to the macrophages. Okay, and then the phagocytosis will bring the pathogen into the cell. It will attach to a lysosome. In that case, it's called a phagosome. The phagosome will the phagosome will get more stuff from the lysosome and actually cause a phagolysosome, and the phagolysosome will eventually break down the bacteria into different parts. Those different parts will be displayed on MHC class 2 molecules, and that way your body will know it will become an antigen presenting cell. It will present it to B cells and T cells about what to kill. Okay. Okay, so the antigen presenting cell will bridge between the innate and the adaptive immune response. That's what that means. Okay, all right. Okay, so after this, the bacterial cells broken down, they will form antigenic fragments. The antigenic fragments will be displayed on the surface of MHC class 2. Okay, and the MHC class 2 will attract B cells and T cells. Okay, once it activates, once the antigen presenting cell activates the B cell or T cell, the B cell or T cell will undergo clonal expansion. Clonal expansion will increase the number of B cells and T cells that bind to that one particular antigen. Okay. Okay. So you can see like clones only bind to their specific antigen. Okay. Okay. So B cells and T cells, okay, both form. I'm gonna skip natural killer cells. Okay, B cells and T cells form effector cells and memory cells. Okay, the memory cells stay around for round 2. Okay, the plasma cells secrete antibodies. Okay, and then the T cells and T cells, the T cells actually do stuff. Okay, the B cells and T cells do different things. Okay, B cells create antibodies. Antibodies are part of something called your humoral response. Okay, it protects the blood and the lymph. T cells are going to be part of your cellular response. They're going to kill virus infected cells. Okay, and regulate other cells. Okay. All right. Okay, so the antigen binds to the B cell. Okay, the B cell will proliferate, they'll make clones. Okay, so here are the clones. The clones also cause memory B cells and effector cells. Effector cells are the plasma cells, which produce a large amount of antibodies. So you've seen the antibodies go up. Okay, the secondary immune response. This is a secondary immune response. Okay, you have the clonal expansion occurs much more rapidly the second time around, and therefore the immune response is much faster the second time around. Okay. All right, antibody functions. Okay, so these are all different antibody functions. Okay, make sure you understand these. Okay. Make sure you understand the difference between active and passive immunity. Okay, you should definitely know what cytotoxic T cells and helper T cells do. Okay, so cytotoxic T cells check everything with MHC class 1 complexes. If it matches, that means that that cell has some component of virus or bacteria that's replicating inside the cell. And if it matches the cytotoxic T cell, then the cytotoxic T cells will kill it by releasing pore-forming porphyrins. Porphyrins perforate the cell, make holes in it, and they release granzymes. Granzymes activate something called FAST to induce apoptosis. Okay. T helper cells bind to immune cells that display antigen in MHC2 complexes, and then they activate other immune cells, B cells and cytotoxic T cells. And HIV destroys the helper T cells. Okay, so this is the helper T cell. Okay, on one end, it activates the cytotoxic T cells. The other end, it actually activates the B cells. Okay, so if I destroy this, then I don't get that, and I don't get that, and therefore I have no immunity, or very little immunity left. Okay. Okay, so T lymphocyte activation. Okay, so remember this is your antigen MHC class 2. Okay, you turn these on, these things turn on, and now they target anything with MHC class 1. If it has MHC class 1, it kills the cell. Okay. Okay, so try to understand what's happening here. Okay, if you understand what's happening here, then you're good to go. Okay. Okay, so this is immune effects to viruses. Okay, so bacteria usually just activate the B cell response is bacteria. Usually, there are exceptions to that rule, but you'll learn that micro, but not physio. Okay, bacteria usually stay in the blood, and therefore they only activate the T cell response. Okay, sorry, the B cell response. Bacteria, B cell response. Okay, viruses, they activate both. Okay, so make sure you understand the differences between a bacterial response and a viral response, because viruses actually enter cells. Okay. So you'll see this side, okay, only with viruses. Okay. Okay. You should definitely understand the difference between immediate hypersensitivity. Okay. Immediate hypersensitivity is mediated by antibodies, and they usually occur within minutes. So think so in microbiology, we call this type 1, type 2, and type 3 responses. Okay. The delayed hypersensitivity is type 4. Okay. So amio, what groups we're skipping, because we covered it in microbiome, we covered it in anatomy, and stuff like that. Okay. Okay. So okay, you should definitely know. So overactive responses. Okay. Incorrect responses, the lack of responses. Okay. So autoimmune disease is an incorrect response, right? So for lack of response, you should know primary versus acquired. So primary is usually a genetic thing, and it's usually congenital. You're born with it. Okay, acquired, you get it from somewhere like HIV. Okay, or radiation can destroy your immune system and stuff like that. Okay. We'll skip this section here. Okay. All right. Hopefully that makes sense to you guys. Okay.